Hello everyone, welcome to QAxon. So in this video, you will learn about the types of skeletal muscle fibers. Under this topic, I will be first covering the two categories of skeletal muscle fibers, and then we shall compare skeletal, smooth and cardiac muscle fibers, and finally we shall discuss the two types of smooth muscle fibers. So here we go. The skeletal muscles can be divided into two different categories, the slow oxidative fibers and the fast glycolytic fibers. Now these two fibers differ both in their metabolism and in their performance. So if you look at slow oxidative fibers, their main source of ATP is oxidative phosphorylation. On the other hand, fast glycolytic fibers are mainly reliant on glycolysis as a source of ATP. Therefore, slow oxidative fibers have many mitochondria because this is where oxidative phosphorylation takes place, whereas fast glycolytic fibers have only a few mitochondria. The number of capillaries is also high in slow oxidative fibers because they bring in the oxygen, whereas capillaries are few in fast glycolytic fibers. The pigment myoglobin, which stores oxygen, is also high in slow oxidative fibers. So these fibers look a little reddish, whereas myoglobin content is very low in fast glycolytic fibers, and so these fibers are a little whitish or pale. The glycogen content obviously is low in slow oxidative fibers and high in fast glycolytic fibers because this glycogen is broken down into glucose, which is then diverted towards glycolysis. If you look at the rate of fatigue, the slow oxidative fibers do not fatigue easily. So their rate of fatigue is slow. But fast glycolytic fibers undergo fatigue very quickly. Finally, the myosin ATPase activity is low in slow oxidative fibers and high in fast glycolytic fibers. So these are the important metabolic differences. There are also some additional differences which we shall now study. So let's discuss the other differences between the slow oxidative and fast glycolytic fibers. So the contraction velocity, as the name itself suggests, is low in case of slow oxidative fibers and high in case of fast glycolytic fibers. There are other structural differences also. For example, if you look at the fiber diameter, slow oxidative fibers have small diameters, whereas the fast glycolytic fibers have a large diameter. Likewise, the motor unit size also differs between the two. The motor unit size is very small in case of slow oxidative fibers, which means that the muscle would have many motor units because the size of each motor unit is small. But the size of each motor unit is large in case of fast glycolytic fibers. Finally, the size of the innervating motor neuron also differs. It is small for the slow oxidative fibers and large for the fast glycolytic fibers. Let's now compare the skeletal, smooth and cardiac muscle. You must already be familiar with the anatomical differences between these three types of muscle. As you can see in this diagram, this is the skeletal muscle. So you can see that the cells are long cylindrical cells called myocytes. They are striated. The nuclei are towards the periphery of the cell, and these are multinucleated cells. On the other hand, when you take the cardiac muscle, the cells are cylindrical, but they are also branched. these regions of branching where one cell connects with the neighboring cell, they're called intercalated discs. This is where the gap junctions are present. The cells are again striated and they have a single nucleus, which is central. On the other hand, if you look at the smooth muscle, it looks rather plain because there are no striations. The cells are spindle-shaped and there is a central single nucleus. Let's now study the differences between skeletal, smooth and cardiac muscle. Now, all these three muscles perform the same function of contraction, but in the body, they serve different functions. So each is tailored physiologically to suit its individual function. So let's now look at the physiological differences between these three categories of muscle. Now we've already studied that the sarcomere is the structural and functional unit of the skeletal muscle. The striations are because of the sarcomere. So skeletal muscle has the sarcomere, but this is absent in the smooth muscle. The cardiac muscle, again being striated, has sarcomere. What about the L tubules and T tubules? We know that L tubules are the longitudinal tubules or the sarcoplasmic reticulum where calcium is stored, and they are abundant in the skeletal muscle. But in the smooth muscle, they are low and they are intermediate in the cardiac muscle. The T tubules, which are inward projections of the sarcolemma, are present in both skeletal and cardiac muscle, but absent in smooth muscle. So what is the source of calcium in each muscle? The skeletal muscle derives its calcium from the L tubules, that is the sarcoplasmic reticulum, whereas the smooth muscle derives it from both the sarcoplasmic reticulum and the extracellular fluid, and likewise the cardiac muscle. If you look at the muscle tone, which is the resting tension in a muscle, the tone of skeletal muscle is nerve dependent, which means if you cut the nerve, the muscle loses its tone. But if you look at smooth muscle and cardiac muscle, their tone is inherent. It is not dependent on the nerve. If you cut the nerve, they do not lose their inherent tone. What about the contraction velocity? Now, skeletal muscle, as we already saw, is of two types: slow oxidative and fast glycolytic. So its contraction velocity is variable. It could be high or low. If you take your eye muscles, which have more of the fast glycolytic fibers, the contraction velocity is very high. Whereas if you take your postural muscles, which have more of the slow oxidative fibers, the contraction velocity is low. But if you look at smooth muscle, the main function that smooth muscles serve often is storage, for example, in the GIT. So we don't need a high contraction velocity. So these have a very low contraction velocity. And likewise, in case of cardiac muscle also, we do not need a very high contraction velocity because the main function of the heart is to pump powerfully enough to generate adequate cardiac output. So cardiac muscle also has a low contraction velocity. Now we already know that the cardiac muscle has pacemaker activity. So pacemaker potential is not seen in skeletal muscle, but it is seen in one type of smooth muscle, which we call a single unit smooth muscle. And it is also seen in cardiac muscle. Likewise, we also know that gap junctions are present in cardiac muscle, absent in skeletal muscle. Again, gap junctions are also present in single unit smooth muscle. Then, if you look at the effect of nerve stimulation. Now we know that all the muscles would respond to nerve stimulation, but remember that skeletal muscles are always excited by nerve stimulation because here the neurotransmitter is always acetylcholine and it is excitatory to skeletal muscle. But when it comes to smooth muscle or cardiac muscle, you could either have excitation or inhibition in response to nerve stimulation. We know that skeletal muscle does not respond to hormones, but both smooth muscle and cardiac muscle do respond to hormones as well, in addition to nerve stimulation. Now, if you look at mechanical stimuli such as stretch, we know that if you take gastrointestinal smooth muscle, it responds to stretch by contracting. But that is not the case with the other muscles. So skeletal muscle does not respond to stretch, but single unit smooth muscle does, and the others do not. And finally, if you look at the molecule that mediates or initiates cross-bridge activity, it is troponin in case of both cardiac and skeletal muscle, but it is calmodulin in case of smooth muscle. Now finally, we will study the differences between the two types of smooth muscle, which is single unit smooth muscle and multi-unit smooth muscle. Now, as the name itself suggests, single unit smooth muscles contract as one unit when we stimulate them. So they respond to a stimulus by contracting as a single unit, but multi-unit smooth muscles contract independently when stimulated. They do not contract as a single unit. We already saw that single unit smooth muscles have pacemaker activity, but this is absent in multi-unit smooth muscles. Likewise, gap junctions are present in single unit smooth muscle but absent in multi-unit smooth muscle. Single unit smooth muscle also responds to mechanical stimuli such as stretch, but multi-unit does not respond. Now, there are examples for both. So we already saw that the smooth muscle of the gastrointestinal tract, the uterus, these are examples of single unit smooth muscle. Whereas if you look at the smooth muscle of the large bronchi or the large diameter blood vessels, these are examples of multi-unit smooth muscle. That brings us to the end of this session. Thank you.